that all have thousands of reactions going on in every single node. And each and every one of these reactions is facilitated by proteins. Proteins drive every single reaction that goes on in your body. And so when something goes wrong with your body, it's most likely because something's going wrong with one of the proteins. And this is why most medications in the form of small drugs actually target proteins to improve their function. The thing is, many times these medications don't work because they're either ineffective at actually targeting the right protein, or they have side effects on other proteins as well. But what if we could actually alter the proteins themselves or use proteins to solve the problem? This is proteotherapeutics, and this was first used with insulin, where they actually started injecting proteins, or the insulin protein, into the body to fix diabetes instead of the medication. The problem is, this doesn't work today because most of the building blocks of proteins, known as amino acids that the body uses, are kind of boring. There are a total of 500 amino acids naturally found in the environment, but our human body only uses 20 of them. This is only 4% of the amino acids, and the thing is, these 20 are basically super boring molecules that all resemble each other very closely and don't have special properties. And this is why in our body, proteins have to fold very intricately and specifically in order to, achieve the functions that they need to. But what if we could actually expand the genomic code to use the rest of the 480 amino acids? Before I get into that, I'm just going to be doing a little bit of review of just genetics. So, as you might know, DNA, or deoxyribonucleic acid, is a type of nucleic acid which is just a really long polymer made up of units called nucleotides. Nucleotides are made up of a ribose sugar, in this case deoxyribose, a phosphate group, and a nitrogenous base, in this case adenine. Now, most of you probably know the 4 letters of DNA, A, C, T, and G. These are the different nitrogenous bases that can be part of the nucleotide: adenine, cytosine, thymine, or guanine. And when you have the 2 strands of DNA line up together, adenine binds with thymine, and cytosine binds with guanine. Next, important process for DNA is replication. So when a cell divides into 2 cells, it actually needs to replicate its DNA so that both cells have the same amount of DNA. So for this to happen, the DNA is first split open by an enzyme known as DNA helicase, after which another enzyme known as DNA polymerase actually slots in the complementary base pairs. So for every A that it finds, it will slot in a T, and for every C, it will slot in a G, and so on and so forth. And once that entire process is done for the entire DNA molecule, you will now have 2 strands of DNA that have the same code. Now, you might be wondering, how do we go from DNA to protein? Well, the first step in this process is transcription. And in this process, one of the strands of DNA, known as the template strand, is actually copied into an RNA molecule. RNA is another type of nucleic acid which just has one single strand, and instead of a T, it uses a U. So just like in the process with DNA replication, the base pairs are copied, creating a new RNA strand known as messenger RNA. This messenger RNA then makes its way into the cytoplasm, into organelles known as ribosomes, where the process of translation is carried out. Here, the mRNA is read 3 letters at a time, or a codon at a time. And once it comes across the start codon, known as AUG, it will start actually synthesizing the protein. For every codon after this, it will bring in the correct amino acid. So in this case, for UAU, it will bring in tyrosine, and for UGC, it will bring in cysteine. And it will continue doing this until it comes across one of the 3 stop codons, which is either UAA, UAG, or UGA. Once it comes across one of these codons, it will release the protein, which will go about and do its function. But you might be wondering, how does this amino acid actually come in? Well, that is another type of RNA molecule known as transfer RNA, or tRNA. tRNA has 2 main important sites in the molecule. The first is the anticodon, which actually recognizes the right codon. And the second is the acceptor stem, where another molecule known as tRNA synthetase will slot in the correct amino acid for that codon. So now, knowing all of that, how can we alter this to incorporate more amino acids that have more useful properties? The first experiment in this area actually tried to repurpose codons, or specifically the UAG codon. In E. coli, the UAG codon is recognized by restricted factor 1, or R1, which makes it code as a stop codon. But in another organism known as Methanococcus janusai, UAG is actually recognized by a tRNA, which slots in a tyrosine instead. So what the researchers did is that first they blocked the action of the restriction factor 1, which meant that UAG no longer coded for a stop codon. And then they put in a bunch of the tyrosyl tRNA synthetase into the E. coli cells, which meant that the UAG now began coding for tyrosine. In later experiments, they changed the structure of the tRNA synthetase to incorporate synthetic amino acids that had been produced in the lab. But there were two main drawbacks with this experiment. The first was that you could only add one more amino acid because you only add in one more codon that you could use. And the second was that you had to recode every UAG in the genome because even though UAG itself is a rare stop codon, there are 83 polypeptides and 314 instances across the genome where UAG is found. And so the researchers had to go through the entire genome and change every UAG into another type of stop codon. The next experiment in this area actually added 2 extra letters. What they found was that the most important thing to be able to use more amino acids was that you had to add more codons. Right now, we have 4 letters, A, C, T, and G, that can go into 3 slots. So 4 times 4 times 4 equals 64 codons. To be able to increase this, we have 2 options. Either we can have more letters per codon. So if we had 4 letters, for example, we could have 256 total different codons. Or we could have more possible letters. Say we had 6 letters, then we could have 216 codons. Even though the first option actually has more codons that it can make, it would require huge changes across the cell. First, we would have to change every single tRNA molecule to actually recognize 4 letters instead of 3. And we would also have to add a letter in the DNA every 3 letters so that it's still coded for the same polypeptides as it did before. So what the researchers did is that they actually added 2 new nucleotides, X and Y, or known as D5-SICS or DNAM. Now, unlike A, C, T, and G, which bond to each other through hydrogen bonding, X and Y bonded to each other through hydrophobic bonding, which they found to be much easier for replication and transcription. The way they actually got this into the cell was first, they injected a plasmid with the X and Y nucleotides into the cell, and a plasmid is just a circular piece of DNA. And then next, what they found was that they weren't actually able to produce X and Y in the cells naturally, so they injected another plasmid with the NTT transporter gene into the E. coli cells. And what this gene did is that it allowed the E. coli to take in the nucleotides from its environment, which the researchers injected the X and Y nucleotides into. Next, the next experiment they did was to test whether the DNA transcription and translation process was working correctly. So they actually altered the green fluorescent protein, or GFP gene, to switch the codon in a non-critical part of the protein from TAC, which actually codes for tyrosine, to AXC. And they injected a bunch of tRNA molecules that had the corresponding anticodon, as well as the synthetic amino acid, PRK. And what they found is that the new GFP protein was synthesized with the non-standard PRK amino acid. Now, this still had one major drawback, and that was the fact that the synthetic nucleotides actually had to be surrounded by the natural nucleotides, which was A, C, T, or G. And the reason was, because hydrophobic bonding is much weaker than hydrogen bonding, if you have multiple of these synthetic nucleotides in a row, they could actually knock out of place and destroy the double helix shape completely. Now, the next experiment in this area actually added 4 extra letters, and they were focusing on 2 main models for genetic information storage. The first was Schrodinger's theory. He said that for a genetic information model to work, regularity in nucleotide size was required to form an aperiodic crystal, which is just any polymer with non-repeating units, which he found to be critical for reliable information storage and transfer. And the second was the Darwinian model, which also found that regularity in size was required, that changes in the sequence didn't actually completely mess up the biopolymer shape, and it could still carry out the process that it normally did, including how it interacted with the enzymes that replicated. So what they did is that they added 4 new nucleotides, S, B, Z, and P, all of which they actually could actually hydrogen bond with one another. And so with this, they actually solved the problem that was found in the previous experiment. And what they tested next was the mutability. So they tested if we change the sequence, does that lead to different functions for the, DNA itself, which it should. And what they found, that even with the sequence changes, different molecules of the new DNA, which they call Hachimoji DNA, had similar structural parameters, showing that the Hachimoji DNA actually acted as a mutable information storage system. The next thing they tested was whether transcription worked, and they found that the T7 RNA polymerase variant known as FAL was able to incorporate the complementary nucleotides for all of the artificial nucleotides. What they found with other variants was that though they were able to copy Z, P, and S, they weren't actually able to find the complementary one for B, but this one was able to do this. And next, they tested whether or not this was able to be transcribed and translated properly. So what they tested was that there's a substance known as FLOR, which when it comes into contact with the spinach fluorescent RNA aptamer, it actually, fluoresces. And what they found was that the first test tube, B, had no spinach fluorescent RNA aptamer. The second one, C, had a Hachimoji spinach aptamer, so the one with the synthetic nucleotides. D had the native one, and so you can see that they had similar fluorescent properties. And then what they predicted was that if we make a change at position 50 in the aptamer, it should actually prevent the fluorescent properties from coming about, and that is what actually happened, which meant that they were able to accurately not only predict how the Hachimoji DNA would be translated and transcribed, it actually showed that it followed the rules that normal DNA followed. And so how can we apply this in the real world? The first application, as I mentioned, was developing proteotherapeutics that can actually solve problems without using small drug molecules that are not always effective. And it can also make new proteins that can accept these drugs more efficiently to make sure that they carry out their function. But other applications include engineering bacteria to carry out whatever functions we might want them to, such as digesting plastic or producing polymers that normal cells can't. And the ones that scientists are most excited about is that this might give us a new idea of what life really might look like on planets other than Earth. Thank you.